Good morning, everyone, and welcome to the next installment of Swanson Reed's new industry-related webinar series. Today's webinar is on R&D in biotechnology and life sciences. My name is Jamie Nickerson, and I'm a technical manager with the Texas Tax Credit Division of Swanson Reed. I'm looking forward to talking to you all about the R&D tax credit for the biotech and life science industries. For the CPAs and EAs taking this course, our field of study today is taxation, and the knowledge level is basic. One CE or CPE credit will be awarded to those who complete the course and correctly fill out the relevant questionnaire. I'll be asking yes and no questions throughout the webinar. You can take time during or after the webinar to answer these questions on Swanson Reed's live webinar page. Please submit your answers to questions 1 through 5 within 20 minutes of the webinar ending to qualify for the free CPE or CE credit. Don't forget to also fill in your contact information and the course evaluation at the end. We'd love your honest feedback. All right, so now the plan for today is we'll start off with an overview of the R&D tax legislation, and then I'll break down the 4-part test to help you identify qualifying research activities in biotech and the life sciences. Then we'll discuss qualifying research expenses in these industries. We'll tie all of this together with a case study and demonstrate how to substantiate an R&D claim through documentation. Lastly, we'll wrap up with a Q&A session at the end of the webinar. And to submit a question at any time throughout the webinar, please visit the Google link displayed at the top of the webinar slides. I'll do my best to go through as many questions as we can with the time we have remaining. All right, so joining me later today is my colleague, Amber Farrington. Amber is a senior manager at Swanson Reed, and she'll be discussing the QRE side of the R&D tax credit. And to give you some background to our company, Swanson Reed is a global R&D tax credit consulting firm. We assist our clients with qualifying for and accessing the financial benefits of federal and state-based research and development incentives, potentially minimizing your tax liabilities. Now, Swanson Reed has helped many companies successfully claim the R&D tax credit. Many of our clients engage in research and development in the biotech and life science industries. We are pleased to share what we've learned from our experience in helping these innovative companies claim the R&D tax credit and access its many benefits. Now, without further ado, let's get into the nitty-gritty of the R&D tax credit. All right, so to start us off, I'll give some background to the R&D tax credit legislation. Then we'll jump into talking about what R&D activities and expenses are eligible in claiming the credit. So the research and development tax credit is a tax incentive for performing qualified research in the United States, resulting in a credit to a tax return. The qualified research does not have to be successful, as the intent of the R&D tax credit legislation was to spur economic activity and encourage companies to engage in risks by way of a tax incentive. The R&D legislation is tailored to a specific jurisdiction, so for the federal tax return, the R&D has to be performed within the United States, since the government is trying to stimulate economic activity within the US and not in other countries where work and research are often outsourced. For states with their own R&D programs, their incentives are limited to R&D that's conducted within their states. So this means that to claim a Texas R&D tax credit, work must be performed within the state of Texas and not Alaska or California, for example. So the R&D tax credit legislation was first introduced in 1981 as part of the Economic Recovery Act under President Ronald Reagan. While it encouraged American companies to engage in R&D, one of the major shortcomings of the act was that it had to be renewed on a year-to-year basis, and this encumbered tax planning. So tax planners did not know whether they could rely on the credit to be available at the end of the year, so they didn't know whether to accumulate data or to take measures to improve their data when conducting R&D. Consequently, the incentive did not get as much attention as it should have. So in December 2015, President Barack Obama signed the Protecting Americans from Tax Hikes Act. This became known as the PATH Act, and it made R&D tax legislation permanent. So tax planners no longer have to worry about whether Congress would renew the legislation, and companies that have sufficient grounds to earn a sizable credit can begin planning for it at the start of a fiscal year. All right, so qualified research activities, also known as QRAs, are the operating activities of a taxpayer that form the basis of the R&D tax credit. Without QRAs, you don't have an R&D claim. It's also important to keep in mind that QRAs must be substantiated and documented, especially in cases of an IRS audit. Now, we will go into more detail about the proper documentation needed for substantiation later on in the presentation. Now, there's a common misconception among the general public that you need to be a large company with full-fledged R&D departments to qualify for the R&D tax credit. On the contrary, to be considered R&D from the perspective of the IRS, your project or projects simply have to meet the four-part test. No matter how big or small your company, no matter the size or structure, if you satisfy all requirements of the four-part test, you could be eligible for the R&D tax credit. The four parts of the test are permitted purpose, technical uncertainty, process of experimentation, and technological in nature. All right, so the first part, permitted purpose, as I mentioned, means the project must have a particular goal or technical objective in mind. The purpose of the project must seek to create new or improve existing functionality, performance, reliability, or quality of a business component. So in other words, you could be aiming to create a new product, improve an existing product, or improving a process. From the perspective of biotechnology, for example, that could mean new product formulations, developing new medicines, new methods of diagnosis, and so forth. The second part of the test is that the project must encompass technical risk or technical uncertainty. So this means that the solution, method, design, and other components were not known beforehand. There was no guarantee that the solution was even feasible, and it was not straightforward to develop. Usually a good clue on whether there's technical risk and uncertainty is if a competent professional in the field finds it difficult to achieve. So for example, if a new engineer who's only been working in the field for less than a year finds a project difficult to achieve, this may not be an indicator of technical uncertainty because it may be challenging due to the individual's limited experience. But let's say if a senior engineer is unable to figure out the solution immediately, then chances are you've got a project with a lot of technical risk and uncertainty on your hands. So this leads us into the third part of the test, that the project must involve a systematic process of experimentation. To overcome those technical uncertainties and risks, you had to engage in experimentation and testing. This means formulating hypotheses, testing and evaluating those hypotheses, exploring alternatives, building prototypes, adjusting designs, and so on and so forth. In biotechnology especially, it is difficult to get it right in one try because you're dealing with complex biological and chemical systems. There are so many interconnected variables and risks that you have to continually test and retest to make sure it all works out in the end. The fourth and final part of the test is that projects must be technological in nature. This means it has to rely on the principles of the hard sciences. So in this case, that would include fields like biology, chemistry, physics, engineering, and many others. It's also important to remember that this criterion makes it clear that the so-called soft sciences do not count. This means projects related to market research, psychology, social sciences, and so on do not qualify as R&D. Now, companies in biotechnology and the life sciences are no strangers to R&D. After all, the quintessential image of a scientist in a white coat conducting experiments in a laboratory, that's the reality of many people working in biotech and life sciences. They're continually looking to develop new scientific methods and processes to advance the scientific and technological knowledge base. Life science and biotechnology encompass many subfields in other industries, ranging from medical technology, agriculture, bioengineering, molecular biology, and several others. As a result, there's a lot of potential for innovation. I'll now go into more detail about each part of the four-part test. All right, so the first part of the four-part test is permitted purpose. This can mean creating a new product, improving an existing product, or improving a process. In the case of biotech and life science, it could be developing complex product formulations, such as developing a treatment for a very specialized illness or condition. Let's say, for example, a company is trying to develop a cure for cancer, or maybe it's to treat asthma. Name any sickness or physical ailment, and chances are there is a lab somewhere working on curing it or treating it. With these treatments, the problem itself is usually complex and requires a new solution. You'd also have to make sure that you can treat the illness effectively while minimizing the risk of negative side effects. So aside from prescription medicines, another possible permitted purpose could be developing treatments for a common ailment found among the general population. These products would not require a special prescription and can be purchased over the counter in drugstores and pharmacies. So, for example, it could be developing a new product to treat dryness in eyes, or perhaps to remove wax in the ear canal, to reduce dryness in the skin, to enhance iron intake, and the list can really go on. With these types of products, You want to be able to firstly treat the targeted ailment effectively, and secondly, make sure it's easy to administer since you're hoping to apply the product for a wide range of people. Now, another area for R&D in biotech is the creation of self-diagnostic tools for medical conditions. Sometimes clinics and doctors' offices are overwhelmed by the number of patients who need to receive a diagnosis, or perhaps testing for a particular illness requires a specialist, but there are a limited number of specialists serving a particular area. So there's a long waiting list. So to reduce these waiting times, which in some cases can be detrimental for the patient, some companies are working on self-diagnostic tools that you could purchase in a CVS or another local pharmacy. These tools will not necessarily give you an authoritative diagnosis per se, but they could provide a more accurate reading of the likelihood that you are suffering from a particular illness, such as cancer or diabetes. These tools would be more reliable than, say, doing what most of us do, going on to Google to determine the causes for your symptoms, as many people are apt to do nowadays. Of course, as we all know, there's always room for improvements. You don't necessarily have to discover the cure for cancer or a new chemical compound to have a permanent purpose. So you could be trying to improve existing products. Maybe you're trying to improve an existing cancer treatment that usually takes 6 months to a year, and instead you want to reduce that time frame to 3 to 6 months. Or perhaps you've developed a water-based solution to treat eye dryness. Your initial solution couldn't work for contact lenses, so you try improving it so that users can apply the solution, even if they were wearing contact lenses. On a larger scale, it could be trying to determine or streamline medical care practice to increase accessibility and productivity. Now, we can all admit that some medical and hospital practices aren't necessarily the most efficient. Perhaps you're trying to develop a software program that can streamline workflows so that there's better communication amongst doctors, nurses, and patients, for example. So evidently, the sky's the limit when it comes to R&D in the biotech and life science industries. All right, so now there's a misconception about R&D, and many people believe that unless you're finding the cure for the common cold or cancer, then it's not R&D. On the contrary, the projects from a tax perspective need to meet the 4-part test, and arguably the most important is the second criterion of the 4-part test, which is that activities must involve technical uncertainty. This is foundational to determining whether a project is R&D. What this criterion means is that there must be uncertainty about the solution, method, and design when starting out on a project. You've identified your technical objective or your goal, but you don't know the best way to achieve that goal. In fact, you're not even sure if your solution will even be successful, and there was potential for failure. Someone who's experienced in the field could not have easily solved the solution. A medical engineer, let's say with 20 years of experience, finds a particular project tricky to develop, typically indicates that the project is more likely to be technologically uncertain than a project undertaken by a fresh graduate. Now, before I get into further details, we now have our first question for CPE and CE purposes, and that is, do you need to be creating a completely new product to be eligible for the R&D tax credit? You can answer in the box to the side of this video on Swanson Reed's live webinar page or by visiting the link shown. I'll give you a few moments to answer. All right, so why were there technical uncertainties? On the one hand, it's because this is a business world where intellectual property is tightly protected, and companies are not trying to share their IP for the world to know, or else they'd lose their competitor advantage. This is true whether it's a pharmaceutical company, a cosmetic company, a biomedical company, and so forth. So let's take, for example, Neutrogena and Garnier are both skincare companies with product lines that include moisturizers, makeup removers, anti-aging creams, eye treatments, and the like. While their products may achieve similar goals, their product formulations and supply chains are likely quite different. The formulation for Neutrogena's anti-aging cream, for example, isn't exactly publicized on the market. Other companies might have tried similar goals, but they failed to achieve them. There could be plenty of reasons for that failure, but again, this information isn't typically what companies would publicize. Other reasons for uncertainties include how there may be some suggestions in academia where researchers have published reports on potential solutions. However, as many can tell you, academia and theory is one thing, and the real world is another. So when you apply these theories into the real world, you might not be able to get the desired results. It could also be that the guidelines that are available in academia or publicly available were too generic for a particular industry. So essentially, then there was uncertainty because there were no precedents to follow. In addition to the practical reasons, there may be restrictions in the existing technology that make it difficult to know how to solve the problem in advance. To give a more real-world analogy, let's say you're trying to build a house in an area that's accustomed to hurricanes and extreme weather conditions. You won't be able to reference building plans of houses built in more temperate climates where the sun is always shining and they never get any storms or tornadoes. So when you're constructing this house in, let's say, the hurricane zone, you don't know what is the best material to use that is durable, water resistant, and can withstand tough winds. You don't know how to design the house to make sure it's flood-proof, structurally sound, and still looks like a normal home and not some sort of bomb shelter. Similarly, in life sciences and biotechnology, certain formulations and products will likewise behave differently in different environments, thus creating a lot of technical uncertainties and risks. It's also important to note that technological uncertainty refers to challenges that are technological and not business challenges. Business challenges include constraints like deadlines and budget. Maybe your company has a particular budget to conduct preclinical trials, but as you carry out the trials, you realize you need to conduct more research and you need to practice more materials to test, but you don't have the budget for it. Or maybe you're designing a custom lipstick for a client, but the client doesn't like the way the cap closes on the lipstick and continually demands that you change it from a round cap to a square one, to a triangular one, and then back to a square one. This is more of a style reason and not for functionality or utility purposes. So while these are understandably frustrating challenges, these alone are not enough to qualify as technical uncertainties and risks. So uncertainties, therefore, are about questions like, is it possible to achieve? Is it feasible? How or what is the best way to achieve it? What is the optimal design? Technical uncertainties will vary from project to project, but I'll try to give some examples to give you an idea of common challenges to the life science and biotech industries. So let's say you're trying to develop technology that can detect certain forms of cancer early on. The technology is something you'd want the average person to be able to operate so they can do it from home without needing to go into a clinic or the hospital or getting professional medical help. Some technical uncertainties you may face with this is not knowing what's the best way to develop a non-invasive method of detection that's safe yet accurate. The risk is that if you develop something that is a bit hands-off, so to speak, for instance, you just need to scan the affected part with light to gather data, it may not be as accurate as getting a more invasive sample, such as a blood or stool sample. You may not know the best way to design the hardware and the software to capture precise readings and provide accurate analysis. So in this case, you're balancing between the goal of effectiveness, such as getting an accurate reading, and the goal of easy to administer, such as a non-invasive method that does not require extensive medical knowledge or expertise. The uncertainty is found in the tension between these two goals, and it's not known beforehand how to resolve the tension. Other possible uncertainties could be trying to develop new product formulations. Let's say you want to develop a hand cream that's over the counter and shipped all across the United States. Some uncertainties for this project include not knowing how to ensure the hand cream can work for different skin types, such as dry, oily, normal, and so forth. It could be not knowing how to ensure the effectiveness of the hand cream in different climates. Someone living in Michigan and having to endure a harsh winter might need a more potent hand cream than someone living in Florida, where the climate is much warmer. Or perhaps you want to extend the shelf life of the hand cream, and you don't know beforehand how to do that when changing one part of the formulation, and can negatively affect the other attributes. In this case, you have a long list of parameters, being shelf life, is it safe, effective in moisturizing hands, and so on and so forth. You have to balance these parameters. If the formulation is too viscous, for example, it might not be absorbable in the skin. If the formulation is too thin, it might dry out more easily, which impacts the shelf life. If it's too potent, it might have an unappealing smell. Again, because of these parameters and conditions, technical uncertainties arise, and you don't know how to overcome these risks and uncertainties at the beginning of your project. You don't know what materials are best to use. You don't know in what proportion these materials should be used. You don't know how long the materials will last, whether they will be effective against the target ailment, if they'll be able to be effective in different climates, if they won't develop negative effects over time, and the list can really go on. Now, a major concern for any biotech and life science project is to make sure that it's safe and reliable. There are so many risks when dealing with biotechnology in the life sciences, of course. So, for example, if you develop a medicine or some medical technology to treat a certain illness, you have to be careful not to unintentionally develop negative side effects, or at least figure out the way to minimize the negative side effects. There are countless examples of prescription drugs that were intended to treat a specific illness, but then turn out to have an unaffected or unintended negative side effect. Safety is paramount to anything in the medical field, and that's why there are important regulations, ethical guidelines, and other standards that these technologies must meet. Now, these regulations create more uncertainties because they add limitations to what you can and cannot do during your experimentation process and product development. So in sum, uncertainties are due to questions related to design, feasibility, integration, and other factors. As you can see, life science and biotech R&D projects can take many forms. Just as there's no limit to the kinds of technical objectives you can have, there are a lot of technical uncertainties and risks that a project will face. These are just some examples, and it's not an exhaustive list by any means. All right, so now moving on to the next criterion of the four-part test known as process of experimentation. Because of the uncertainties and challenges you faced at the outset of the project, you had to eliminate these uncertainties by undergoing a systematic process of experimentation. This means things like testing different options and alternatives to determine what solutions work and what don't work. It's also important to note that the work you're doing doesn't necessarily have to be successful in order to qualify. In fact, if you tried something that didn't work or failed, it demonstrates the need for experimentation to eliminate uncertainty. In fact, it's quite common in R&D to engage in experiments and find that what you had in mind initially to start out with didn't turn out the way you intended. Certain problems you initially identified might have turned out to be even more complicated than you thought. And so you had to go back to the drawing board and modify your initial designs and plans. Again, that's okay because it shows that there was the elimination of uncertainty and therefore can be considered R&D. Experimentation involves coming up with a hypothesis or an idea of what you're trying to achieve. You then investigate the scientific knowledge base to identify potential solutions and to determine proof of concept, basically assessing whether a design concept is feasible. You then try developing different designs and formulations. You test these designs by developing prototypes, which you further test and monitor. You may make modifications to the prototypes based on the test results. So in the case of pharmaceutical development, you would test the drug prototypes through preclinical trials, making sure the product is safe before it can be tested on humans in clinical trials. Then you'd have different phases of clinical trials from phase 0 to phase 4, thus carefully monitoring the effects of the drug. So throughout these trials and tests, you may discover that you have to make modifications to your original formulation. And again, that's okay. It's all part of the experimentation process. After all, the clinical trials, you may start scaling up the treatment from small lab batches to full-scale manufacturing. So during the scale-up stage, new challenges might arise. For example, you need to increase the proportion of materials for full-scale production. You might find that the materials start to separate out of the formulation during the scale-up, so you have to add other ingredients to stabilize the formulation, and this will require further testing and experimentation. Now, aside from drug product formulations and the like, companies are also increasingly introducing software and computer science into their biotech and life science projects. The experimentation process would involve similar patterns of development, but would also include things like computer simulations, lab testing, software development, and so forth. These activities would also qualify as part of the four-part test. So to summarize, the process of experimentation part of the 4-part test is just that, experimentation. Now, this is different from random trial and error where you're kind of spontaneously throwing things together in hopes of getting some sort of result. With experimentation, you have to have a set goal in mind, the technical objective, and some hypothesis on how to achieve that goal. Your hypothesis might be wrong or it might be right. Either way, you need to test that hypothesis and evaluate that testing through a process of experimentation. It's a systematic process, and usually a good indicator of that systematic process is if you were documenting the progress for the work. So for example, you might have background research, literature reviews, project records, testing protocols, documents recording the results of testing, testing protocols, and things of that nature. All right, so lastly, the 4th criterion of the 4-part test is that the project must follow the principles of the hard sciences. In the case of biotechnology, it's pretty clear that projects in this field will fulfill the criterion of the 4-part test. This could mean anything from the life sciences like biology, chemistry, and physics, to the more applied sciences like engineering. All right, so now to recap, to qualify for the R&D tax credit projects, projects must meet all of these parts of the test: permitted purpose, elimination of technical uncertainty, process of experimentation, and technological in nature. All right, so this leads us into our second question for CPE and CE purposes, and that is, do activities need to meet all of the criteria to qualify for the R&D tax credit? You can answer on Swanson Reed's webinar page or by visiting the link shown. I'll give you a few moments to answer. All right, so who is eligible for the R&D tax credit? Here's a simple pie chart so you can visualize how the industry is spread in claiming the R&D tax credit. You can see that there is not any one field that has a monopoly on this incentive. Manufacturing, including biotech production, is one of the largest of the claimants. This is typically because companies in this industry are continually developing new product formulations, whether it's new pharmaceuticals, drugs, cosmetics, food recipes, and the list can go on really. You can also see that professional and technical services are growing industries that are taking advantage of the credit. Professional services include doctors, medical professionals, hospitals, and so on. Again, all industries and companies of all sizes can qualify for the R&D tax credit, as long as they can satisfy the IRS's 4-part test. All right, and as you can see, here we have our next question for CE and CPE credit purposes, and that is, are all industries able to access the R&D tax credit? Again, you can answer in the box on Swanson Reed's live webinar page or by clicking the link below. And again, I'll give you a few moments to answer. All right, so now that you have a general overview of the eligibility criteria for QRAs, I would like to welcome my colleague Amber to tell us more about the second part of the R&D tax credit. Amber?
Thank you, Jamie, and hello, everyone. Thank you again for joining. So as Jamie mentioned, I will move us into the next part of the R&D tax credit, which is qualifying research expenses, or QREs. Documenting and substantiating these expenses will be the most work our clients have to do when claiming the R&D tax credit, and Swanson Reed will do the rest. This is also probably the most important part of this presentation as far as the taxpayer is concerned, because we need these expenses in order to calculate and prepare your R&D tax claim. QREs are the operating expenses of a taxpayer that form the basis of the R&D credit. Like I said, QREs must be substantiated and documented, for example, by way of receipts, invoices, or properly documenting and labeling expense and use of in your company's chart of accounts. It is very important to keep in mind that you cannot make estimates on costs, as the IRS does not accept estimates or educated guesses. Nothing will undo a claim quicker than not documenting the expenses that you're claiming. So there are three major categories of qualifying research expenses, being payroll, supplies, and contracted research services. So payroll or R&D labor expenses include scientists, biotechnicians, principal investigators, analysts, whoever happens to be working directly on the R&D project. These personnel may not only or always be working on the R&D project. For example, a medical engineer could only be spending half of his day working on R&D, so only 50% of his payroll could be claimed as an R&D expense. The amount of labor claimed as an expense to the project should be proportionate to the amount of time spent on R&D. So like I said, R&D labor expenses can only include direct labor and first-line supervisors to the project. For example, you cannot claim the salary of the CEO unless, of course, he or she is working directly on the project, which does happen, but of course is not as common. Indirect labor can be included, but only if they're performing specialized work on the R&D project. For example, this could include data collectors or specialized personnel that come in specifically to clean or sterilize the test site or equipment. That being said, here we would exclude general janitorial staff, payroll clerks, security, basically anyone who performs these general support jobs. And any payroll or R&D labor expenses claimed must have been for staff or personnel closely associated with the R&D project. So the second major category of qualifying research expenses is supplies. So the same as with labor, these have to be supplies that were directly consumed in performing the R&D. The definition of R&D supplies, according to the Internal Revenue Code, means any tangible property other than land or improvements to land, and property subject to depreciation allowances. The reasons for these exclusions are that land can never be depreciated, and that the taxpayer will already be calculating and receiving depreciation benefits from its capital assets. The IRS does not want to give benefits for capital assets on both the side of depreciation and on the side of using it for R&D supplies. As the benefit is already being received through the complex process of depreciating capital assets, the IRS has created this blanket rule that if it's a capital asset that's depreciated, it is restricted from being claimed as part of R&D. So supplies should ideally be entirely consumed or expended in the tax year being claimed. Furthermore, it is ideal that supplies are used up entirely in the R&D process with no scrap or resale value. This is because if you use a supply for the R&D project, but then find additional use for it elsewhere, then you get into an issue of trying to allocate how much of the asset you used towards R&D. Okay, so we already touched on this a bit in the previous slide when discussing the definition of supplies, but just to reiterate, supplies excludes capitalized equipment as the benefit is received exclusively through the depreciation process and cannot be brought into calculating QREs for the R&D claim. Depreciation is typically limited to capital equipment with a service life of at least 1 year. Overall, trying to synchronize allowances for credits with depreciation schedules is an effort in futility and would likely result in a tax offset greater than the basis or original cost of the equipment, which is why the IRS will not allow capitalized equipment to be used as a QRE for the R&D claim. Okay, so the last category of qualifying research expenses are contracted research services. These are companies or personnel outside of your company that assist you in your research, with the best examples being universities or colleges and testing labs. They could be contracted to do additional specialized research, anything directly related to the R&D project. It's important to avoid claiming services using conventional knowledge, such as services from electricians and plumbers, that could be seen as enhancing the taxpayer's plant and equipment for multiple purposes. The takeaway here really is that R&D is not a cash cow for general plant renovations. Okay, so we have another question for the CE/CPE credit, and it is, must all R&D be conducted by in-house staff to be eligible for the R&D tax credit? Again, you can answer on Swanson Reed's live webinar page, or if you're watching this from YouTube, you can visit the link shown on the slide here. Okay, moving on to carry forwards. So just like net operating losses, R&D credits can be carried forward 20 years and back 1 year until they're fully expended. Carry forwards are calculated as the difference between the credit calculated at Form 6765, which is the Federal Research and Development Claim Form, and then Form 3800 Part 2, Line 38, which is where you would claim what you want to use in that year. So carry forwards are as good as cash. A lot of people like to have it all up front, but if you can't use it now because you've earned such a large credit, lucky you, because you will have it for future years. All right, so in summary, we have learned that qualifying research activities are the operating activities of a taxpayer's R&D claim. QRAs must meet the requirements of the four-part test. Qualifying research expenses are the operating expenses of a taxpayer's R&D claim. It is very important that taxpayers identify QREs that are complete and R&D related. And lastly, R&D credits can be carried forward 20 years and back 1 year until fully expended, and they are as good as cash. Okay, so now that you have the background to QRAs and QREs with some examples, Jamie will break it all down with a case study that will explain the QRAs and QREs of a fictional company. Jamie?
All right, thank you, Amber. So we have Derma Incorporated, a company that specializes in over-the-counter products that treat a wide range of medical ailments, from symptoms of the common cold to skin-related conditions. One of the company's products is a multi-step eczema treatment kit consisting of a day cream, a night cream, and a mask. In 2014, the company wanted to improve its existing eczema treatment kit by reducing the number of products needed in the kit to just the night cream. So in other words, it's going down from 3 products to just 1. So in order to qualify for the R&D tax credit, Derma needed to determine the eligibility of its proposed R&D activities. The qualified research must meet 4 main criteria, known as the IRS's 4-part test. All right, so an R&D tax credit specialist helped Derma determine its qualifying R&D activities, and that included development and testing of a night cream that did not require a day cream and a mask in order to effectively treat the symptoms of eczema, experimented with different formulations and ongoing testing of the stability, effectiveness, and safety of the product, lab testing and clinical trials of the product to evaluate performance of the product, monitoring the performance of the product during scale-up. So how does that meet the 4-part test? Firstly, Derma was seeking to improve an existing product. In this case, it was to reduce the number of steps needed to treat eczema from 3 stages to just 1. The company found that the multi-step process was too cumbersome for users because it required multiple applications throughout the day and the week. So with this new project, the company is cutting back on production costs while also enhancing the effectiveness of its product by making it easier and simpler to use. As an over-the-counter product, the night cream also had to be safe to apply on any skin type without any negative side effects, and that the night cream just had to have a reasonable shelf life. All of these objectives meet the permitted purpose criterion of the 4-part test. Secondly, Derma's project meets the technical uncertainty criterion because there were many unknown factors and risks involved. For example, the company's original product involved 3 steps. By going from 3 to just 1 step, you don't know for sure if the new product is going to be still be as reliable and effective in treating eczema. So perhaps in the original treatment, the night cream was useful at preparing the skin to better absorb the day cream, and the day cream was the main step where eczema was treated more aggressively. Perhaps the mask was only used once a week as a protective layer. Now, with its new product, Derma has to ensure that the night cream is able to still do what the day cream and the mask did. So, however, the best way to achieve this isn't known. You don't know if you could combine all the existing formulations, meaning adding the day cream and the mask formulations to the night cream to make a super formulation, if you will. Or you would only modify and enhance certain properties of the night cream. You also don't know if the standalone night cream would still be effective for a variety of skin types, among other uncertainties. All right, so to eliminate uncertainties, Derma had to undergo a systematic process of experimentation. It would investigate and identify possible solutions and alternatives. It would test and evaluate these different approaches and formulations according to safety, reliability, functionality, performance, and consistency. As it was testing and experimenting, it would discover new issues and challenges, and therefore it would have to modify the formulation based on these test results until an adequate solution was determined. So, for example, Derma may be trying to investigate and develop a new substance or material that has a positive impact on treating eczema. So this new substance has to be more potent in treating eczema than materials previously used in the day cream and the mask as well. Maybe there's no natural ingredient that can do this, so the company has to experiment with developing a new substance from scratch. From its research, and after several iterations, Derma developed an ingredient that achieves what it wants, but when it adds this ingredient to its existing night cream formulation, the ingredient starts separating in the formulation because of how the ingredient reacts with the other ingredients. The company has to undergo further testing and experimentation to stabilize the formulation. Maybe it adjusts the different portions of ingredients. It might decide to eliminate some ingredients, or it might discover that it has to add another ingredient to stabilize the formulation. After a formulation is determined, the company then has to engage in preclinical and clinical trials to make sure the product is safe and effective. The results of the trials will also have an impact on the original formulation, as new problems are identified and new modifications will have to be implemented. Okay, so lastly, Derma meets the 4-part test because it relies on the principles of the hard sciences, in this case, chemistry, physics, biology, and engineering. All right, so to sum up the QRA side of this study, Derma meets the IRS's 4-part test because firstly, it was trying to improve an existing product. Secondly, the project encompassed technical risks and uncertainty. Thirdly, the company had to engage in a systematic process of experimentation to eliminate those uncertainties. And lastly, the company did indeed follow the principles of the hard sciences. Therefore, it is eligible for the R&D tax credit. All right, so now that we've established that Derma does meet the 4-part test, the next step is to identify qualified research expenses. As we covered earlier in the presentation, there are 3 categories of qualifying research expenses, being labor, supplies, and contracted research services. Your company may have all costs across all categories or just in one. In this case study, Derma had expenses in all 3. Labor is typically one of the largest expenses for companies, so being able to claim these expenses for the R&D tax credit is actually quite beneficial. In Derma's case, they employed a team of 3 full-time scientists who worked directly on the R&D project 100% of the time. Derma is able to claim 100% of labor costs in calculating their R&D tax credit, which, as any company can tell you, is quite a sizable amount. The second QRE category is supplies. They also had supply costs relating to chemical reagents, lab equipment, and other costs 100% related to their R&D project. 100% of supply costs are eligible in calculating the R&D tax credit. Derma contracted out part of their research to a medical laboratory to conduct clinical trials. Only 65% of contracted research costs are eligible in calculating the R&D tax credit. All right, so similar to any tax credit or deduction, Derma had to save business records to substantiate their R&D claim by outlining what it did in its R&D activities, including experimental activities and documents to prove that the work took place in a systematic manner. So Derma saved the following documentation to substantiate its qualifying research activities, those being project records, including things like lab notes, literature reviews and background research, records of change, testing protocols, results and records of analysis during testing, including preclinical and clinical trials. The company also saved the following documentation to substantiate their qualifying research expenses: records of resource allocation, usage logs, staff timesheets or W-2s, invoices, receipts, chart of accounts for R&D supplies, and 1099s for contracted research. This is by no means an exhaustive list. As you can see, documentation is very important to have on hand in order to substantiate an R&D claim. It's not as arduous or difficult as you might think. Many companies already have these files on hand. While it's not required to submit documentation, it is very useful to have on hand in the case of an IRS audit. All right, and here we have our last question of the day. Is documentation required with submission when filing a federal R&D tax credit claim? Again, you can answer on Swanson Reed's live webinar page or by visiting the link shown in the box on this slide. I'll give you a few moments to answer. All right, and that concludes the presentation side of this webinar. Now we will move into the Q&A portion of our webinar where we will answer your questions. So I'll jump right into it. So our first question today is, I'm not exactly inventing the wheels, so how do I know if my work is R&D? Thanks for the great question. We know that in popular understanding, R&D is something that's incredibly groundbreaking and creating something completely new. However, from the perspective of R&D tax legislation, it's more about encouraging American companies to engage in economic and technical risks and innovation. Keeping that in mind, you can determine if your work is R&D using the 4-part test that I had mentioned earlier. I know I had mentioned it quite a few times, but just to refresh your memory, the 4-part test includes permitted purpose, technical uncertainty, process of experimentation, and be technological in nature. If you can say yes to all 4 questions, it's more than likely that you have a qualifying R&D project. All right, so on to our next question. My project wasn't able to make it to market. Would it still qualify for the R&D tax credit? Yes, it could. The beauty of R&D is that your project doesn't have to be successful to qualify for the tax credit. If you attempted to do a project and there were technical failures, you can communicate those to the IRS when you file for the tax credit. If your project encompassed all the elements from the 4-part test, it will still qualify for the R&D tax credit, marketable outcome or not. In fact, that's the value of the R&D tax credit, since you can start benefiting from your expenses even before it went to market. All right, so our next question is, are R&D tax credits transferable? Yes, they are. They are just like any other asset or debt with a company. If a company has debts, those debts are inherited by the buyer of that company. R&D credits are effectively an asset and will be passed on to the buyer of the company. All right, and on to our next question. What kind of documentation do we need to send if we file a claim? All right, so for payroll, if you're a calendar year taxpayer, W-2s or a payroll register will do the trick. If you're a fiscal year, we will need a payroll register because it needs to line up with your operating or fiscal year. With supplies, the ultimate evidence is an invoice, but Swanson Reed does not need copies of all invoices. They can simply be summarized in an Excel spreadsheet. This is actually ideal. However, in the event of an audit, you need to be able to locate those actual invoices. So just make sure if you're claiming the costs, you have the documentation to back it up. And lastly, for contracted services, you'd simply need copies of 1099s. All right, now there are several questions, but unfortunately we are now out of time. I truly appreciate everyone taking the time to participate and ask such great questions. So thank you so much. We are out of time now, but Swanson Reed's next step is to help you identify the qualifying research expenses that your company has incurred over the last few years and put a dollar amount on what the net financial benefit would be to your company on claiming the credit that you have already earned, but just have not claimed yet. Now, we would like to prepare an initial R&D tax credit study at no cost to your business. This initial study will help you determine exactly the financial benefit is going to be for your company, the LLC members, or S Corp members. We want to put an estimate together in real dollars and cents so that you and your CPA can decide if it's even commercial or worth your while to take the next step in the claims process. I want to thank you all for your time today. Please reach out with any additional questions, and we look forward to preparing the no-cost R&D study so that you can decide which tax years, if any, make sense to claim. For all CPAs or EAs taking this course, please remember to submit your questionnaire within the next 20 minutes after ending the webinar. Thank you all again, and bye for now.